Sunita Madam and Rao sir for giving me this opportunity to deliver my first lecture in first week only. And respected participants, good evening everyone. Today I will be talking a little bit less than 1 hour about gene editing technologies, and that too will concentrate mainly on CRISPR-Cas9 technology, how this technology is better than the other existing technologies. So when we say genome editing, so since the discovery of DNA and later several projects like Human Genome Project, everything, so one of the major goal of genetic engineers was how to make specific changes in DNA. Suppose when we take a genome is very, very large. Human genome is approximately 3 billion base pairs. We want to change a small portion of a gene inside that genome. It is like you are looking for a needle in a haystack. So, and you want to make changes in one particular region, only one specific change. So, how do you achieve that? So, that was one of the main goal of people, genetic experts, they were trying to. Suppose when we see a lot of genetic mutations, mutation happens in particular gene only. So, you want to alter that gene only and not anywhere in the gene. So, to achieve that, people were looking for better tools, better enzymes, etc. And in that particular search for technologies, people have come up with over the years from 1970s onwards, several things have come up. So people have discovered restriction endonucleases. So that was like one of the landmark discovery for genetic engineering and so on. But restriction endonucleases, what do? They cut everywhere in the genome. Cutting is very random. So, but you want to make a specific change inside any one of the genes. During this process, people have developed certain technologies, gene editing technologies such as zinc finger nucleases, or in short we refer to them as ZFN. Then the second technology that came up, which is slightly better than ZFN, is TALENs, transcription activator-like effector nucleases. And better than these two techniques, final one is the CRISPR-Cas9 technology. So we'll just take a brief look at how these technologies work, their advantages and disadvantages. So as I was telling, so people were looking for enzymes which can do specific changes. And in that process they discovered endonucleases, restriction endonucleases. But majority of endonucleases, they have a very specific cutting site. Suppose if you take BAM-H1, it has GG80CC. It cuts only at that location. And if you take EcoR1, AAGCPD, it cuts only at that location. That could be present anywhere in the genome at thousands of places. So it cuts everywhere. You don't want it. Then during that process, they discovered a peculiar type of enzyme called as a class of enzymes, type 2S enzyme. And this has an interesting mechanism of action. One of the example is FOC1 enzyme. This FOC1 enzyme, it has two domains. One is a DNA recognition domain and second is a catalytic domain or cleavage domain. So this particular DNA recognition domain has a specific recognition site. It recognizes only this region, GG80G. Whereas this catalytic domain or cleavage domain, it doesn't have any recognition. So whatever is there adjacent to this particular recognition sites, it cuts there. It cuts there and leave a 4 to 5 base pair overhang, 5' overhang. So that is the mechanism of action of FOC1 enzyme. So people took this enzyme as an interesting enzyme, and what did they do is, and it is a dimer. One more thing is, it works only as a dimer. When two units, similar units come together, then only it can act. One unit will be on one of the strand and the second unit will be on the bottom strand. So now they engineered this particular enzyme, removed the DNA recognition part, and they took only the catalytic domain, which is a dimer. So what did they do next is, and we want specificity. Now we have a domain which can cut anywhere in the gene. And now we have to bring in a second portion which is highly specific. You want to cut somewhere in particular gene, you have to bring in the specificity. So this specificity was brought about by second part called as zinc finger domains. So zinc finger domains are some of the proteins which have the capacity to go and bind to a DNA. And these zinc finger domains they are nothing but proteins only. We can engineer these particular proteins to bind any sequence on the cell. So we need to engineer this particular domain. Once we engineer that domain, and we have to fuse it with the fork 1 catalytic domain or fork 1 cleavage domain. So now zinc finger domain binds to region of our specificity, and the adjacent fork 1 cleavage site cuts the DNA. So thereby you can inactivate the gene, or you can do many other changes wherever you want to do. So how does that work? Let us see. So these zinc finger domains are DNA binding domains. They can bind to DNA. And people have studied in human genome itself, there are nearly 1400 zinc finger domains are there. And one of the class which we discussed is, among these 1400, nearly half of them have a class, they belong to class called as C2H2. So why they call C2H2 is, this is the entire motif. Motif means domain, the sequence. X could be any amino acid. X could be any amino acid. Cis is C. Another cis is C2. So two cis are there, two histidines are there. So C2H2 is a type of zinc finger protein. And this X3, X12, it could be any amino acid, doesn't matter. So this is one of the zinc finger domain. Now we can engineer these amino acids in between, change slightly the amino acids, and we can make them bind to any DNA on this side. So how we have genetic code, right? We have genetic code, there are 64 codons are there, and each codon codes for a particular amino acid. Now people have done same thing, reverse engineering, and this one, each particular zinc finger domain, one domain, can bind to one codon. So when a zinc finger domain binds to DNA, one domain can bind to 3 bases or one codon. So that is how they do. So next what people do is, so this is how a single domain, and they have a structure called as, it belongs to beta beta alpha group. This alpha helix is the one inside this portion which will attach itself to the DNA. Which will attach itself to the DNA. Now this is one codon, base 1, base 2, base 3. So based on, suppose you want to engineer a particular domain which you want to bind to G. So what to do is, inside there, arginine 6, you have to, 6th amino acid should be arginine, then histidine, arginine. If we have this particular combination, they go and bind to G. So the next slide, if you see, we can clearly see that. See each domain here, this is one zinc finger domain, second, third, fourth. So we can engineer this particular domain to bind only to GCC. How we do is, we go back to previous slide. So these amino acids can be changed to bind to any codon on this side. So all 64 codons we can bind. And already people have sequence. If you want to particular codon, you want to engineer a zinc finger domain, it is already there. Just we have to get it synthesized or make a protein out of itself. So already everything is existing. So we need not engineer the newly. So already engineered motifs are there which can bind to anything. So now thing is, as I told you, fork 1 it works only as a dimer. So we need to engineer not just one zinc finger domain. We need to engineer an upper strand as well as lower strand. So the chance of two enzymes coming together is very rare. Means, suppose these are the sites we have engineered, they can bind only here. And chance of these two binding somewhere else and cutting is very rare. Because here 4 3s are 12 and 4 3s are 12. Totally 25 base pairs are there in total complete this region, present in this gene. Chance of this region present elsewhere in the genome is very rare, negligible. So it is a highly specific method of making editing genome. So once you engineer these zinc finger domains, it doesn't matter. Wherever fork 1 they come, zinc finger domain binds and dimerizes, they cut the DNA here. They cut the DNA here and then leave a 5' overhang. So similarly, by adjusting these zinc finger domains using already existing zinc finger structures, we can cut any DNA on that. and edit only that particular thing and not any other things. So, and another thing is, so this is very good technology, specificity is very high. It cuts only the region where we are intending. But thing is, engineering a separate protein is like a tedious process as well as a costly process. So people come up with a next another they discovered after zinc fingers, they discovered a second class of DNA binding proteins called as TACE, transcription activator like effector nucleases. So even this technique was tested with a lot of tools and it has successfully worked. Only thing is it is tedious process as well as costly process. So people have discovered the second technique, this is Talens. They discovered another class of DNA binding proteins called as TACE, transcription activator like effector proteins. So these proteins were discovered in a bacteria plant pathogen called as Pseudomonas. So this particular protein, it is also a DNA binding protein and it has inside this protein there are several repeats are there. These repeats, when we see a single repeat, it contains nearly 34 amino acids and of these 34 amino acids all are conserved. All are conserved except this 12 and 13 which is HD. H is histidine, D is aspartic acid. So these are the two amino acids. So these, whenever that particular repetitive domain has HD, that region it binds to guanine base. It binds to guanine base. So therefore, we can engineer only this region, the 12 and 13 amino acid in the entire tail, and we can make it bind to any region on the earth, any DNA sequence like this. So people use, suppose if you want your tail to bind, like there is a DNA sequence GGATCC. Now how do you engineer is, suppose there is a base A is there in your DNA, then inside the tail you have to keep Ni. N is asparagine, I is isoleucine. So you need to engineer your tail protein such that at the 12th and 13th position you will have asparagine and isoleucine. Suppose if your DNA contains cytosine base, then you need to engineer your particular tail protein 12 and 13 amino acid as histidine and aspartic acid. So just by changing two amino acids only, you can engineer the total complete tail protein as you wish, so that it binds to only your gene of interest, your region of interest. And same thing again here they use fork only, fork 1 cutting domain. So like this HD binds to cytosine. See here, HD is histidine and aspartic acid. So when it binds to DNA, it binds only to the cytosine base. So like that we can engineer all the tail domains to bind to our specific DNA sequence. Here NN can bind to guanine. NN it binds to only guanine base. So we can engineer total tails, tail repeats. Each repeat can bind to only one base. Each repeat can bind to only one base. So this is how when we see side view, tails they surround the DNA double helix, and this is the top view. When you look DNA from top, this is how they appear. So this is how tail repeats will look like. So each repeat can bind to one base. And you need to change only like this. If you have 20 tails, the 20 tails will bind to only the 20 base pairs only. So we have to design a separate tail for bottom strand, a separate tail for upper strand. And now fork 1 they dimerize and cut in between. So that is how we can engineer ZFNs as well as tails to make cutting anywhere possible. So perfect specific genome entry can be made using this particular two techniques. So okay, these two are good. Thing is, these are tedious. Engineering these particular proteins, you need to engineer the complete domain, zinc finger domain as well as tail domain, and you need to get the complete thing synthesized, make a protein out of that. So now it becomes a tail protein and fork 1. They have to dimerize inside the cell and then make it. So it is a more tedious process. What if there is a technique which doesn't involve any of this protein engineering? You don't have to engineer no protein. So people were looking for even better technique. That is how when they came across the CRISPR-Cas9. So the advantage or popularity of CRISPR-9 comes from its simplicity. Simplicity as well as its dynamic nature. Very easy to manipulate. There is no protein engineering. Very simple technique. How does it work? Let us take it. See, though these are well established and people have tested them in real scenario, problem is protein engineering is time consuming, expensive. So to get the whole thing designed, getting it synthesized, making them into a protein and delivering it to the cell, it takes a tedious process. And for each target, in the sense, to cut one DNA, you have to make two engineered proteins, one on upper strand and another on lower strand. So that is also a tedious mechanism. And suppose if you deliver it to 100 cells, the chance of editing happens in very less fraction. Only a minor fraction of cells will get the real genetic change or genome edits. And another disadvantage is, suppose you want to, in the same cells, you want to make edits at 2 to 3 places. Make edits in 2 to 3 different places in the same cell. So multiplexing can't be done with this technique. It becomes a very difficult process. So all these disadvantages were covered by one single technique called as CRISPR-Cas9. So we will take a look at how this CRISPR-Cas9 technique works. So CRISPR is a abbreviation for a thing called as clustered regularly interspaced short palindromic repeats. So CRISPR in essence it stands for clustered regularly interspaced short palindromic repeats. So everything is there in the name only. Clustered means present adjacent to each other in several repeats. Regularly interspaced. So these repeats are present at uniform lengths and interspaced short palindromic repeats. So palindrome is something which reads same on either side. So it is a family of DNA sequences which are found in prokaryotes such as bacteria and eukaryotes. So people have discovered as early as 1970s only these regions, but they didn't know exactly what did they serve for bacteria. So people understood them later as the bioinformatic technologies and everything progressed. So later on, during early 2000s, people discovered that these CRISPR sequences, they are derived from bacteriophages. So bacteriophages are viruses which attack bacteria. So when bacteria get attacked by a bacteriophage, so suppose when we get an infection, what does our body do? It raises an immune response. And instead of raising second immune response in future, it develops a memory. Our immune system develops a memory of plasma cells and other T cells. So when there is a second infection, the time taken for second response is much shorter. Similarly, bacteria also have evolved a type of adaptive immune response. Suppose once they get infected by a bacteria, they remember that bacteria. And when there is a reinfection from similar or same bacteria, they remember that memory and then use it to attack destroy the bacteria. We will discuss how it works. So these CRISPR sequences or CRISPR system is present in maximum 50% of bacteria we know and 90% of archaea bacteria. So CRISPR is used to detect and destroy DNA of bacterial viruses. So it is used for their own protection. They protect themselves from bacteriophages using this CRISPR loci. How it works, let us see. And not only that, during subsequent infections also, they protect themselves from this. So it is one of the antiviral defense mechanism of bacteria. So it was discovered in E. coli genome in 1987, but people only really understood their function in 2007. So when they tried to match this sequence using bioinformatics tools, they found that these regions have similarity with bacteriophage gene. So then they understood maybe when there is a bacteriophage infection, some of the DNA of bacteriophage is taken and placed inside the CRISPR loci. CRISPR is a region of their genome. Where lot of these repeats are present. So whenever there is a bacteriophage infection, a portion of their genome is taken and placed there. So next what they do is, when there is a reinfection, So this is how it works. When the bacterial cell is infected with virus, it captures small pieces of the virus DNA. First thing it does is, whenever there is a bacteriophage infection, and it tries to take a portion of its genome and insert the viral genome into their DNA in particular pattern to create segments known as CRISPR RNAs. So in the CRISPR loci of their genome, it takes the portion of the bacteriophage genome and places it inside the CRISPR loci. Like that it will have 10, 20, 30, 40, any number. Depending on the bacteriophages it encounters. So this CRISPR array allows the bacteria to remember the virus. So once it inserts a portion of bacteriophage genome, it remembers the virus. How it remembers, let us see in the next stage. So if there is a virus attack, the bacteria produce RNA segments from the CRISPR array that recognize and attach specific region of viral DNA. So what happens is, you would have known a technique called as RNA interference. RNA interference. Where suppose if there is a RNA and another RNA goes and binds, there is a double-stranded RNA hybrid. There is an enzyme called as dicer, it goes and destroys that. Similarly, there is also a system like that. Whenever there is a viral genome inside a bacteria, immediately what happens is, this bacteria, it transcribes the CRISPR loci. Whatever loci it has, it transcribes it. Now a portion of that CRISPR loci, it goes and attaches itself to the viral genome. Because of the complement array. Once it attaches to the viral genome, there is a double-stranded hybrid is there. Once this double-stranded hybrid is there, an enzyme called as Cas nuclease, CRISPR-associated nuclease, they bind to those hybrids and destroy the viral genome into pieces. That is how they protect themselves from the bacteriophage or viruses. So once the hybrids are formed to the viral specific region of DNA, bacteria use Cas9 enzyme to cut the viral DNA into pieces, and thus it disables the virus. So this is how it works. So when there is an initial infection, what happens is, a portion of viral DNA is taken and placed inside the CRISPR loci. Like that several are there from different bacteriophages. So once there is a CRISPR loci, a portion is there, what happens is, suppose if there is a second infection from the same bacteria and its genome enters the bacterial cell, immediately what it does is, it transcribes the entire CRISPR loci. So wherever this CRISPR matching is there with the viral genome, they go and make a hybrid variant. So there are two portions are there here. This one which double-stranded or binds, make a complementary strand is called as guide RNA. Guide means it goes and attaches to specific sequence of bacteriophage. This is guide DNA, guide RNA, and this part is called as tracer RNA. Means it forms a specific hairpin loop. This hairpin loop recruits the Cas9 enzyme. It recruits the Cas9 enzyme. Now Cas9 enzyme cuts the viral genome into pieces and it protects the cell. So now people have discovered its mechanism. Now they use this technique and to manipulate any genome, any gene sequence on this. Just what they need to do is change only this guide RNA. See, tracer RNA will remain intact. Whatever is there original from bacteria, they keep it same. Just what we need to do is to change this guide RNA to the sequence you want. So now what it does, guide RNA takes the tracer RNA and goes and sits on the wherever it finds specifically inside the gene. Once it sits there, tracer RNA recruits Cas9 and cuts the region at specific locations. So thereby you can disable the gene, or through homologous recombination, defective gene can be replaced by a real gene or a functional gene. So that is how people do that. So it's same thing only. And apart from that, this Cas9 or tracer RNA, it has a specific requirement called as PAM. PAM is a specific small sequence. Tracer binds only at PAM sequences. It doesn't bind anywhere on genome. So we need to look for sites which have PAM. PAM is protospacer adjacent motif. So that is also another requirement, but it is very easy, simple. How we can do that is, let us see. So these are small examples. cRNA or CRISPR RNA is guide RNA. Guide RNA which goes to specific genome location and binds to it. Tracer RNA is which is the double-stranded hairpin loop, and that recruits Cas9 enzyme. So in real location, real life, or which we use technology, we refer to it as single guide RNA. Hence, this RNA contains both guide as well as tracer RNA. So in CRISPR-Cas9 technology we use word called as single guide RNA, and Cas9 enzyme it doesn't matter. We all know it is the endonuclease makes a cut. So this is how it works. This is the PAM, protospacer adjacent. So this is a small region called as PAM. PAM site means each bacteria has a specific PAM site. So generally the CRISPR-Cas9 technology we use is from a bacteria called as Streptococcus pyogenes. Streptococcus pyogenes which cause phospholipid infections. So it has a protospacer motif called as GGN. N could be any amino acid. GG presence is very easily available. If you take a small gene, 1000 base pair gene, presence of GGN is nearly 10 to 20 sites in billion. Very simple sequence. Because the PAM availability is more, people are using Streptococcus pyogenes. There are other Cas9 which have a different protospacer requirement. Since Streptococcus has simple protospacer PAM region, people use this one more frequently. So we need to engineer our guide DNA adjacent to the PAM motif. So wherever suppose we want to make a change in, let us say, beta globulin, or somebody is having albinism. Albinism, there is a single amino acid change. So you search the entire melanin gene or beta globulin or anything. See where these PAM motifs are. You design the guide RNA adjacent to the PAM motif. So this is our target DNA. Let us imagine target DNA. You found a PAM sequence. So now you design a guide DNA adjacent to PAM motif. So this is our guide RNA, and this part is tracer RNA, which is naturally present in the bacteria. We don't change that because it forms a specific hairpin loop, and it is needed for Cas9 function. So now once it is bound, Cas9 comes and makes a cut exactly 3 bases adjacent to the PAM site. So DNA is cut. The cut is double-stranded break. Both strands are cut. So once we make a cut, any double-strand breaks are there in the genome. Cell tries to repair the breaks. So it does by 2 mechanisms. So one is non-homologous enjoining, another is homologous recombination. These are the 2 methods cell employs. Non-homologous enjoining is what it does is, as soon as it finds a double-stranded break, it tries to fuse them. And when it joins the double-stranded breaks, 2 things will happen. Either there is some deletion or there is some addition. If it finds nearby any DNA there, it inserts there. So their insertion happens. Suppose by joining it doesn't find any DNA nearby, it deletes a portion of the double-stranded break and rejoins. So in non-homologous enjoining, 2 things will happen, either insertion or deletion. When these 2 things happen, gene becomes dysfunctional. There is a gene knockout. Gene doesn't function anymore in the cell. There gene knockout has happened. The second mechanism what we do is, once double-stranded breaks are made, we also deliver whichever region you want to insert, we will also deliver that into the cell. And since that particular insert is present in high concentration inside the nucleus, what this repair mechanism does is, cell repair mechanism, it inserts that region using a homologous recombination. So defective gene will be replaced by a functional gene. We deliver it in a plasmid. Plasmid will go there, and from a defective gene it exchanges and delivers a functional gene. So any genetic mutations in individuals can be corrected using CRISPR-Cas9 technology. First you send the CRISPR-Cas9, it makes a double-stranded breaks. Next what we do is, along with that we have to send the functional gene in a plasmid. Now using homologous recombination, the defective gene will be replaced by a functional gene. So that is how one of the most promising technologies is CRISPR-Cas9. You see how simple it is when compared to zinc finger nuclease and tail mechanism. There we need to engineer lot of proteins. Two proteins need to be engineered in zinc finger as well as tails also. Here there is no protein engineering. All you need is single guide RNA of 20-30 base pair pairs. And another requirement is PAM-SIC, which can be located so many places inside a gene. So that is the simplicity and dynamic nature of the CRISPR-Cas9 technology. So its history means these sequences were discovered as early as 1987, and later they were also discovered in 1992 from Archaea bacteria, primitive bacteria, and it later, because of their repetitive nature, they named it as CRISPR by this particular scientist, Musica. And in 2002, adjacent to the CRISPR loci, they have discovered several genes, Cas nucleases, close by only. So Cas nucleases were also discovered in 2002. Later, after the Human Genome Project and everything, our advancements in collection of so many sequences, what happened to is they found similarity between CRISPR regions and bacteriophage DNA. Somehow they thought bacteriophage genetic fragments are incorporated into the CRISPR loci. And 2007 their real function was understood. How they help protect against the bacteriophage infections. And using this in 2012, people have first time they have genetically engineered or they made a genetic manipulation using this CRISPR-Cas9 technology. And these two people, Emmanuel Charpentier and Jennifer Doudna, they made the first experimental proof for genome editing in eukaryotic cells using this technology. So for their contribution in 2020, they were awarded Nobel Prize in Chemistry. So same thing. All CRISPR-Cas9 technology immediate requirement is a small guide RNA in your gene of interest where you want to make the edit. So once guide RNA goes and binds to the region, and since tracer RNA is there, that edit structure, it recruits Cas9 enzyme, make the double-stranded break. So these are the normal things. Once breaks are there, cell uses its own DNA repair mechanism. So two things as I told, non-homologous enjoining and homologous recombination. So in this particular mechanism, when we don't deliver anything, simply what happens is either insertion or deletion happens and there is a gene knockout. So we would be listening gene knockout, mice, gene knockout, bacteria like that. It means where gene doesn't function. What gene knockout does is, suppose we have lot of genes are there. We don't know function of majority of them. You want to know what exactly a gene does to a cell, you simply knock out the gene and see how cell behaves. Whether a cell can live or which metabolic pathway is affected. So these things can be known. So gene knockout experiments are very, very crucial in understanding the function of a gene. So gene knockouts can be easily made using CRISPR-Cas9 technology. Simply what you do is make a guide RNA, deliver Cas9. It cuts the DNA, there ends the matter. Insertion or deletion happens, gene knockout. Suppose there is a defective gene. Suppose somebody is having hemophilia, or somebody is having any genetic disease because of base mutations or things like that. You want to swap a defective gene with a functional gene. Now what we need to do is, in addition to making CRISPR-Cas9 cuts, we need to deliver a functional gene in the form of a plasmid. So once Cas9 makes a double-stranded break in the genome, their breaks are there. Immediately what it does is cell repair mechanism. It uses the DNA present on the vicinity and using homologous recombination, it swaps out a defective gene with a functional gene. See the beauty of this technology. Some families, they have genetic disorders and it kept on passing from generation to generation. And suppose if you correct in one generation this genetic defect, next generation onwards it is safe. So these genetic defects we can correct in somatic cells or we can even correct in germline cells. When we correct them at the germline cells in early embryonic development, last stage like that, and child who is born is free from that particular genetic defect. So that is the potential of this particular technology. So So it is same thing, homologous recombination. So moment there is a double stranded break, if we deliver something like a, we need to create a homologous regions. So once homologous regions are there, what happens is they bind here and then deliver the outside itself. You can do anything using this particular technique. And the advantages is it is much faster than the remaining two process, much cheaper, more accurate, more efficient than other genome editing techniques, and much simpler also. So it has been tested in several fields like pathogen diagnostics, even treatment of pathogenic diseases, treatment of HIV, and the functions are unlimited. So people have used CRISPR-Cas9 technology to develop a transgenic animals, transgenic mice, transgenic several animal models and bacterial models. Several GMOs are also created, and people have used at germline level, very early in development only, to use CRISPR to correct the genetic mistakes. So it has potential to treat cancers and other genetic conditions like progeria, beta thalassemia, sickle cell disease, hemophilia, cystic fibrosis. Anything can be corrected using this technology. And not only that, we can use same technology to control bacterial infections also. So when there is a bacterial infection, you use CRISPR to control the bacterial virulence factors, bacterial antibiotic resistance mechanisms. Thereby you can control the bacterial infection also. So when we see the real world scenario, this is some of the examples where people have created using CRISPR technology a particular tomato variety called as Sicilian Rogue High GABA variety, SRH3. So this particular tomato has a metabolite called as gamma aminobutyric acid, which is very much essential for brain development. So it is a amino acid, kind of amino acid, non-proteinaceous amino acid. It is not part of the proteins, but it is an amino acid needed for brain development. And it has 5 times more quantity than a regular tomato. So simply what they did is, this is the original genome. There is a particular gene called as AAD, which suppresses the GABA production. Simply what they did is, they knocked out this particular AAD gene. Now there is a high production of GABA. So this negatively suppressing gene was arrested. Thereby, plant tomato produces high amount of gamma aminobutyric acid. And second example is, this you would have listened probably, a Chinese genetic engineer, he created genetically engineered babies using CRISPR-Cas9 technology, and he made these babies HIV resistant. So at the germline level only, what it is, he corrected the embryos or zygotes at a very early stages and made these embryos, HIV resistant. We all know HIV, when it infects a class of immune cells which have CC5 receptor, CCR5 receptor. So what he did is, in the embryo, he knocked out the CCR receptor itself. When there is no CCR5 receptor, the person doesn't contact HIV. So that is how he made a HIV resistant genotype individuals. So, and later there was a severe backlash, and he was slapped with a severe fine as well as imprisonment also. Means these are ethical questions, we can do anything, but how much we can do is up to the scientific community and up to us. So sometimes what happens is, one of the limitations is off-target effect. Means other than the target, sometimes it can go and cut some other regions. So that thing, it was there at the when people introduced CRISPR-Cas9, but with advancement and progress in recent times, off-target effects reduced to very minimum. And once people started using Streptococcus pyogenes Cas9, it reduced to very, very little. There are no off-target effects are there. And another limitation is PAM, protospacer motif agglutinin, right? That is the only requirement. We need to see that that motif is there in gene where we are cutting. Except that requirement, nothing else is needed. Suppose if you take a small gene, you can see the number of places where you can see the protospacer motif. There are a lot of places where GGN can be found. This is a very commonly occurring three-stretch nucleotide. So even if you take any small gene, you can find tens or more of protospacer motifs. And you need not search. Once you give your gene sequence, there are a number of softwares are there. They can select you the best guide RNA. And technology that's it. So these are all the softwares, bioinformatic tools. Just feed your gene sequence, you can design hundreds of guide RNAs. So you can use the guide RNA, whichever could be best, and you can perform the CRISPR-Cas9 technique. Either you can do gene knockout or we can do gene replacement. Anything can be done using this particular technique. So with this, I conclude gene editing technologies, zinc finger nuclease, clearance, as well as CRISPR-Cas9 technology. Thank you, sirs and madams, for this opportunity. Any questions, please? చాలా ఈజీగా కొనుక్కోవచ్చు మనము Cas9 జస్ట్ ఈరోజు ప్రిపేర్ ఉంటేనే ఎంజైమ్ ఆల్ నీడ్ ఇస్ మనం గైడ్ RNA డిజైన్ చేసుకొని ట్రేసర్ RNA కలిపి మనం సీక్వెన్స్ ఇస్తే కదా 2-3 రోజుల్లో వస్తుంది. వాళ్ళు గైడ్ RNA జస్ట్ సింగిల్ గైడ్ RNA సెన్సర్ చేపిస్తారు. అండ్ అది మనం డైరెక్ట్గా యూజ్ చేసుకోవడం. సింగిల్ గైడ్ RNA Cas9 మిక్స్ చేసి సెల్స్కి ట్రాన్స్ఫర్ చేయాలంటే ట్రాన్స్ఫర్షన్ రీజన్ కూడా చాలా చీప్. పీపుల్ డూ లైక్ ఎంత ఈజీ అంటే కదా మనం రోజు ల్యాబ్లో అబ్జర్వ్ చేసినట్టు అంత ఈజీగా చేసుకుంటున్నాం. చాలా యాక్సెస్ ఈజీలీ యాక్సెస్ ఉంది. ఐఏఎస్సి సిఓ లాంటి స్టేజ్లో ఎవ్రీ ల్యాబ్ యూజ్ యూజింగ్ CRISPR-Cas9.
డాబాలో ఉండే టమాటాలో ఉండే డాబాను మనం యూజ్ చేసుకుంటాం. అంటే డైజెస్ట్ అయిపోతుంది కదా.
అంటే చిన్న అమినో యాసిడ్ వేస్తారు, అది మనకి అబ్జర్వ్ అవుతుంది. ఈవెన్ మనం ప్రోటీన్స్ తిన్నా కానీ అమినో యాసిడ్స్ బ్రేక్ అవుతాయి కదా. బాడీ యూజెస్ దోస్ అమినో యాసిడ్స్ డైరెక్ట్లీ ఇంటూ ప్రోటీన్ బిల్డింగ్. జాబా చాలా చిన్నది మేడం, అది డైజెస్ట్ అవ్వదు. వెరీ స్మాల్ అమినో యాసిడ్. అండ్ ప్రోటీన్ అయితే కనుక డైజెస్ట్ అయ్యి అమినో యాసిడ్ అవుతుంది. ఐ థింక్ ఇట్ ఈస్ మోస్ట్లీ అవ్వదు. బాడీ క్యాన్ అబ్జర్వ్. చిన్న మాలిక్యూల్ డైజెస్ట్ అయ్యేది సార్, విటమిన్ లాగా అబ్జర్వ్ అవుతుంది.